Genetic engineering is done in a lab, and farming is done in a field. So how do we get DNA from under the microscope to seeds under the dirt? I'm Nick Saik, and this is how engineered food gets from a lab to a farm. First things first, you've got to engineer or alter the genetics of the plant. Now you could do that with various kinds of breeding. Traditional breeding and hybrid breeding as examples. And there are other kinds as well, and some of them are so sophisticated that you wouldn't be wrong to call them engineering. But breeding is different from engineering because when you breed things, you typically get offspring, seeds. With genetic engineering, scientists are working on a much smaller scale than seeds even. When you engineer a gene, you don't automatically get a seed. You're probably left with a cell, actually. You've got to get from a cell to a seed. Now, we've touched on how scientists actually make changes to genes in other videos, and we'll get in more depth about other ways they do that in future videos too. There are many ways to actually engineer or change genes or insert genes into a plant. Point is, that isn't even technically the hard part most of the time. Changing genes in one cell is pretty easy, actually. The hard part is getting the one cell to be a whole plant again. Once a scientist has actually done the modification thing, then they've got to make a visit to the
tissue culture transformation lab.
Okay. Tissue culturing is almost always part of the process of getting genes and cells back to seed form. In Kenya, I was walked through the process of tissue culturing by Dr. Simon Gichuki and two members of his lab team, Penina Mwisa and Elizabeth Kamani. I think I got the last one right.
So after the materials leave the laboratories, this is where you can do like your transformation work. Right. And before they go out into the greenhouses, they are processed here. In the laboratory, they create the genes and they put them in the cells. But here now, these cells are put in the plants.
Right. So we're on track then. By this point, the genes have already been put into a cell, and that cell can actually sprout shoots, growing a plant from a single cell rather than a seed. With most species, you end up with a fertile plant that produces engineered seeds. So we're done, right? We've gone from a lab to seeds. Next step, the farm. Thanks for watching. Not quite. Culturing still plays a big role even after that initial seed-bearing GE plant has been created.
Any plant part can be subcultured for the purpose of obtaining plant root or leaf or stem or axillary buds.
That first generation of GE seeds from that one plant is a pretty small yield of seeds, right? I mean, the seeds came from a one-off plant. At this point, they're kind of rare. Culturing allows scientists to make copies of that first plant from its tissue rather than waiting for it to produce seeds. Saves time, big time. And besides that, scientists likely have a ways to go before they actually arrive at a stable trait that works consistently. Oftentimes, the plants that do really well growing in a cell in a petri dish aren't actually great at growing in the field. Introducing that trait into other varieties is a pretty crucial step in the evaluation process of that trait. Scientists have to make sure their work is stable over many generations. Simply put, scientists make changes to the genes of plants, and through culturing, they are able to reproduce and check their work. Culturing is essential to the process of genetic engineering. It's almost biblical in a way. As the old story goes, God cultures Adam, literally from mud, and then tissue samples a rib and cultures Eve, I guess, from that, bone of my bone and all that. So what does this all actually look like? Well, I'm sorry to say I didn't get any shots of the actual cells or shoots forming, but I did get to see the aftermath. This is a grow room. It's filled with cultured plants. These didn't start as seeds. They started as a piece of a plant. It's kind of cool.
This is cassava.
This is cassava.
Yes,
there's some cultured cassava. Is this genetically modified cassava?
No, this is just a normal cassava.
Just a normal cassava?
Yeah, normal cassava. And also on the other side we have vanilla. This one is vanilla.
Vanilla. And there's some cultured vanilla. But fun fact, did you know that vanilla comes from an orchid flower? Specifically, the only kind of edible orchid. I learned that in an orchid lab in Hawaii, and we'll do an episode on that in the future. I really like these plants in jars, and growth rooms in general seem like bright, happy places, but the plant's journey doesn't end here. They won't stay in a jar forever. So this is our biosafety level 2 greenhouse. Okay.
And in this greenhouse, what you saw in the labs, eventually you bring them and test them in the greenhouse.
Right.
So that before we go to the farms, we are sure that what we expected to get is correct.
So just for the public's own understanding, you're not inventing a trait and then just throwing it at farmers.
No.
You make sure.
Yes, before we actually even go to, not even the research farms, we have tested it in the laboratory, as you saw in the laboratory. Then we test it in our either greenhouses or our greenhouses.
Testing.
Then finally we test them in the field.
Okay. GMO food, genetically engineered food, is not something that begins its day under a microscope and then ends its day in the dirt, in a field. The process is intense. And the engineered plants are tested again and again at every stage to ensure things are going well. Like Simon mentioned, from the greenhouse, eventually the plants get to a research farm like this. Then the plants would get to a seed production farm to propagate, make more seeds, and more testing. And then finally, they would find their way to a farmer's field like this. The distance between the engineering of a gene and the consuming of an engineered plant is huge. The road plants have to take to cover that distance is arduous, and it's filled with checks and balances in testing. Well, I'm Saik, and that's how GMOs get from the lab to the field. Thanks for watching.